Warm greetings, students. In today's class, we are going to learn about the type of microscope and we are going to mainly focus on electron microscope. Here, we can simply divide the microscope into light microscope and electron microscope. Under light microscope, you have various types, and under electron microscope, you have SEM, TEM, and STEM, which is a combination of these two. Here, to see a quick review on what is the simple difference between the light microscope and electron microscope, the source is the major difference. When we compare these two, here we have light as a source to view the specimen, whereas here we have electrons to magnify the final image. Under that, we have many magnifying lenses which condenses the light to pass through the object so that we finally get a magnified image. Here as well, we have a set of magnetic lens which condenses the scattered beam of electrons through the specimen, and it is being visualized, or a magnified final image is projected on the photographic film or a fluorescent screen, and then it is visualized. So here we are going to learn, a complete insight on electron microscope. Here, as we told, we are going to use an accelerated beam of electrons in order to produce a final magnified image, which has been projected onto a phosphorescent screen or phosphorescent screen, because the accelerated beam of electrons would harm the eye. So we are going to focus it onto the fluorescent screen and then visualize it. This is how a typical electron microscope looks in the laboratory. The major or important parts of electron microscope is listed down here. We have an electron gun, which is nothing but the donor of accelerated beam of electrons, which is nothing but the heated tungsten filament, which is capable of generating a high intense beam of electrons under the influence of electric voltage. And we have set of electromagnetic lenses. Here we have the condenser lens. From the source, we have the accelerated beam of electrons coming up, which has been focused onto the condenser. The role of condenser is to condense all the beam of electrons to pass through the specimen. It will not allow any sort of electrons to divert from there. It will allow, or it condenses all the accelerated beam of electrons to pass through the specimen so that we get a better magnified image. And then we have a very important lenses. They are nothing but the objective lens and the ocular lens seen in other type of microscopes as well. Here we have the objective lens, which is capable of producing a intermediate magnified image, whereas we have the projector lens, which produces the final magnified image. We have the specimen holder, which is nothing but extremely thin film of carbon or metal. And then we have the final imaging that is being done with the help of the fluorescent screen. As I told you, we cannot visualize it directly. Under electron microscope, we are going to learn about two type of microscope, that is the SEM and TEM analysis: transmission electron microscope and scanning electron microscope. Here in transmission electron microscope, we have the electron gun, of course, the heated tungsten filament. We have anode, which is capable of attracting all the negative charges, and we have the condenser lens, which is capable of condensing all the beam of electrons that is coming from the donor. It will reach the specimen. It is how it condenses or forces all the accelerated beam of electrons to pass through the specimen. Here we have the objective lens, which is capable of producing the intermediate image. Here we have the intermediate lens, which is again a set of magnetic lens, which is capable of producing a better magnified image. We have the projector lens, which produces the final magnified image, which is being projected onto the fluorescent screen and can be visualized at the end. And on the other hand, we have SEM, that is nothing but the scanning electron microscope. Here we have the electron gun, as well we have the anode, which attracts all the negatively charged, that is the electrons, which has been passed through the set of magnetic lenses, which is of course a objective lens and a ocular lens, that is your projector lens. And it also has a backscattered electron detector. As the electron hits onto the specimen, there are some amount of electrons which will be repelled back, or which will be triggered back, which has been detected by the backscattered electrons. And we have the secondary electrons. As soon as the electrons hit onto the specimen, there is a set of secondary electrons that are produced, which in turn produces the magnified image. So here we are going to detect the secondary electrons as well. So the difference between the backscattered electron and the secondary electrons should be understood, and then it is being projected onto the photographic film. So this is how the final image is being produced in both segment 10. But to see its differences, or see its principle, or see its unique feature at which situation, which electron microscope we choose, is very, very important. In case of a study of any sort of external topography or external structure or external composition, external chemical bonding, scanning electron microscope is extensively used, because it is the one which is highly capable of studying the external topography of the specimen or the composition of the specimen. Here to speak a little bit about its principle. Here you understood that it is going to produce secondary electrons. When the beam of electrons strike onto the specimen, they discharge some amount of tiny electrons, shiver a tiny drop of electrons. These electrons are nothing but the secondary electrons. If I say a magnified image is being produced, it is because of the production of secondary electrons. These secondary electrons are now made to fall onto the scintillator film, which produces flashes of light. These flashes of light are in turn amplified, and then the final magnified image is being produced. So this is how a magnified image is being produced. First, the secondary electrons are produced, which is made to incident onto the scintillator film, which causes a flash of light. These flash of lights are now converted using a beam of electric current, and then these signals are now transmitted into a photographic film. So there are many other sort of electron source that we can use, which can be tungsten filament, of course, which is most efficiently used because of its cost or because of its convenience in usage and all that. Also we have field emission gun, which is also used, and also we have the cerium hebaboride cathode, which produces 10 times brighter electron sources. So here we have the other components or structure of a cell. If we speak about, you have the magnetic lens, of course, which is made up of your objective lens and ocular lens. Ocular lens is also called as projector lens, and you have a vacuum chamber. As we are going to use a highly accelerated beam of electrons, the dust particles in the air shouldn't distract them or divert them. So we are going to maintain a vacuumated condition, so that no dust particles would intervene into the way of the electrons. And here we have the sample chamber or the stage, which can hold the specimen, and we have the computer, which means we can produce digital magnified images which can be saved as a soft copy. And then we have the secondary electron detector, of course, and we have the backscattered electron detectors and the power supply here. What are the main applications of scanning electron microscope? It is usually highly used in studying the external topography. So in turn, it is used in life science laboratories, biology laboratories, gemology, medical sciences, also forensic laboratories, examination of the surface contaminants. It is also used in many sort of crystal studies, bond studies, structure studies, external topography, chemical composition, bond structure. All this can be studied in case of scanning electron microscope. They have their own advantages. Production of 3D structure or topographic film is being formed, which is of super magnification, where great details can be visualized. Very less time is required. Minimal sample preparation is required. It produces, of course, a digital data, easy to operate under a proper training. Also, it has its own disadvantages. This is nothing but its cost, and it requires its own room setup, and a vacuumated condition has to be maintained. So, of course, it is again contributing to the expense. Again, it requires a high voltage requirement of the cool water in order to operate the same. Of course, a proper trained person is required. It is limited in studying the solid. As I told, it scans the ultrastructure, the external topography alone. So this is well known to study the solid components alone. And it also has a risk of radiation exposure as we are working with highly accelerated beam of electrons. So just to switch over to temp, to just to bridge the gap, here we can understand that the SEM works under the influence of scattered beam of electrons. Again, the transmission electron microscope works under the influence of transmitted electrons. Here, in case if it is the secondary electrons which gives rise to the magnetic magnified image, here we have this transmitted electrons which contributes to the production of the magnified image. Electrons are directed towards the sample and it passes and it produces the final image. But here, in case of scanning electron microscope, scattered electrons are focused mainly. Here in TEM, we can analyze the sample as a whole, but in case of SEM, we can analyze the sample bit by bit only. Here we have the explanation for transmission electron microscope in a very brief manner. It has been used as a very powerful methodology to study even a very single particle, especially in case of nanoparticle study. Transmission electron microscope is highly used. It produces over 2 million times better magnification in case of light microscope. To compare the light microscope and the TEM, here we have, as we know, as the resolving power increases, the better the magnification, the wavelength of the light decreases. But in case of TEM, the better the resolving power, the wavelength of the electrons in turn increases. So the magnification is much more better because the wavelength of the electrons are not being reduced like the light in light microscope. That is why the magnification is very, very effective. And here also, as we saw in the construction, they have their own set of magnetic lenses which consist of objective lens and ocular lens, which finally produces a polychromatic image. When I have to speak about the advantages of TEM, it produces over 2 million times better image when compared to the light, of course, and it is used for the nanoparticle study. Especially in nanotechnology, it has been highly used. It produces a very efficient, greater detailed magnified image. It is a permanent image, of course, a digital image, and it is very easy to be operated. But it has its own disadvantages, like it is expensive and it is should be handled with care. Proper training is required. The preparation of sample is a bit tedious process. The use of chemical fixation, hydration, or embedment may affect the sample. Maintenance has to be paid attention. The person who operates should be trained properly. They are extremely sensitive to any sort of vibration or electromagnetic movements. So where we place the apparatus or where we place this stem or where it is set up, the room should be free from any sort of other radiations. It produces a monochromatic image unless they use a fluorescent screen at the end of the visualization. As I told you, under the influence of fluorescent screen, we can produce a polychromatic image, but if no fluorescent screen is used, we can produce monochromatic image. To understand its applications, it is used for study of all the organisms, microscopic organisms. Of course, internal details can be understood where bacterial flagella, bacterial plasmid, or bacterial cell organelles can be understood. As I told you, scanning electron microscope studies only the topmost layer of the external topography, external composition, or external bonding can be studied. Transmission electron microscope has the ability to penetrate deep inside and study the cellular structure as well, cellular components as well. That is why the study of cell organelles can be done using TEM. in a comparative analytics in the cellular organism, or to study or to work on its composition. Again, TEM is of greater use. As I told you, nanotechnology, nanoparticles can be studied using TEM, and it is used to detect or identify the fractures or damaged microparticles which can be repaired easily. So these are the advantages, disadvantages, applications, differences of SEM and TEM analysis. To conclude the presentation, here we can come to a conclusion that SEM and TEM are equally efficient, but which we choose at what a sample study is very, very important. Choosing the right type of electron microscope will help us to get a better data. Thank you.